The overall goal of this procedure is to isolate pancreatic mesenchymal cells for their gene and surface marker expression analysis.
This method can help answer key questions in the pancreas development field, such as what cues are expressed by mesenchymal cells and how do they influence epithelial development.
The main advantages of this technique are that it is relatively simple and fast and that it allows a good yield of sorted cells.
Begin by depositing the internal organs in a culture dish containing HBSS and placing the dish under a stereo microscope. Detach the liver and kidneys to expose the pancreas, using fine forceps to remove the pancreas from the stomach, duodenum, and spleen. As each pancreas is collected, transfer the tissue into a conical tube containing freshly prepared digestive buffer and place the tube on ice. When all of the pancreata have been harvested, transfer the tissues to a 37 degrees Celsius heating block for 30 minutes with agitation at 700 RPM, manually inverting the tubes 3 to 4 times after 15 minutes.
If large pieces of pancreas are still present after the first half of the digestion, invert the tube every 5 minutes for the last 15 minutes and incubate the tissue for an additional 5 to 10 minutes as necessary.
At the end of the digestion, place the tubes on ice and add 10 milliliters of cold HBSS to each sample. Collect the cells by centrifugation and resuspend the pellets in 2 milliliters of fresh PBS. Next, filter the cells through 30